Okay, good morning, everybody. And so we're going to carry on our discussion about plants. We're going to see how far we get. Remember, I'm not going to examine you on material which we have not covered in these classes, okay, even no matter what your syllabus says. But if you're going to be writing MCATs or something like that, you might want to read over the extra chapters that we don't cover. Okay, so here's one of the characteristics of the seed plants. We've discussed quite a few of the characteristics of the seed plants, the seed in particular, the structure of the seed. But here's another characteristic which has evolved in the seed plants, and that is the development of a fruit in the angiosperms, in the flowering plants. You do see things that look a little bit like fruit in some of the gymnosperms. You remember the ginkgo fruit that I told you that smells so terrible. That is not really a true fruit. It's actually a spore, which has been enormously enlarged and made fleshy to be attractive to animals or whatever, to try and disperse the seed. But a true fruit consists of a mature ovary and sometimes other flower parts as well around it. And the most common way a fruit is formed is by enormously thickening the ovary wall after fertilization has taken place. And we'll see a picture in a minute. I'll show you. That is very typical of fruits that we eat. But sometimes other parts of the flower itself may become involved in constructing the fruit. The fruit serves very often as a protective mechanism. The fruit may, for example, may be bitter or sour for a very long time and unattractive to animals or birds, whatever, while the seed is developing. And then once the seed is mature, the fruit sweetens or somehow develops an attractive quality and gets eaten. Along with it, seeds get eaten, the animal moves away, bird flies off and poops out the seed somewhere distant from the parent plant. In other words, fruits are a way of dispersing seed, and they're a key evolutionary development because it leads to an interaction between those animals which feed on the fruit and disperse the seed and the plant itself. It's one of the things that drives the development of certain kinds of fruit. Now, the fruit is not always fleshy and attractive for eating. The fruit may also be dry, and it may be adapted in such a way that it aids dispersal. The fruit may, for instance, have hooks on it, which catch on animals' fur, and they take it far away. The fruit may also have wings and be blown by wind. There are various modifications of the fruit. All of them are designed to help disperse the seed away from the parent plant. There are many reasons why that's advantageous. First of all, the parent plant may be large and might shade out. If it just dropped its fruit and dropped all of its progeny below it, very soon, when those germinated, they'd be shaded out by the parent plant. And the progeny in that case would be competing with the parent for nutrients, water, and everything else. It's disadvantageous. Much more advantageous to disperse your seed far away. Here are typical fruits, and this is one of the easiest to understand. Here is a tomato, and you can see that this is an ovary. Go back to that diagram of an ovary, and you'll see that here are the ovules. The seeds here are formed from the ovules. They're all here, and this is the ovary wall that has become thick and fleshy. Of course, this is a little bit deceptive because many of these things have been bred. Okay, human beings have bred them to be particularly fleshy, particularly edible. This is a grapefruit. It's the same idea. It's the ovary enormously expanded with the seeds in the middle. And each of these, in fact, is an ovule. Each segment here is an ovule. The nectarines, the stone fruit, are a little bit different. This here is the ovary there, forming this very hard fruit, this very hard seed in the middle. And this is why these are referred to as stone fruit. The fleshy part is actually the base of the ovary, which has expanded up and enclosed the entire ovary to form a fruit. That's what I meant when I said other parts of the flower can become involved. Various parts of the flower can contribute to formation of a fruit. Fruits here, the nuts, this in fact here is the ovary wall, and this is ovule there inside. And they may also be adapted, of course, you know. They may develop elaborate hairs, be able to be dispersed by wind. And in some cases, in very specialized cases, for instance, in the orchids, there is no fruit at all. In fact, the seed contains virtually no nutrients for the embryo at all. The seed just consists of a very small particle, which is a tough outer covering, the integument, and then the embryo inside. And the idea there is that those are so small that they can actually be blown, almost like spores. They can be blown away. But that's a very specialized adaptation. In most plants, they have some elaboration of the fruit. There are more. This is what I meant by having some of them produce these hooked coats, but they may also have wings to be blown away and this sort of thing. But typically, the fruits are attractive to animals, attractive to birds. The fleshy part is nutrient rich and is also tasty. And in eating the fruit, the animals, birds, whatever, they eat the seed as well and transport it away. Okay, so here's the really amazing thing about the flowering plants. In the gymnosperms, as we talked about before, the process of fertilization was relatively simple. We had a megaspore that divided by meiosis to produce a haploid egg, and that haploid egg got fertilized by a sperm. Now, that division in the megaspore, in the gymnosperms, becomes elaborated on enormously in the angiosperms. And I've put this is a diagram which is not initially in your PowerPoint set. I'll make sure that it is there for you because this explains it really clearly for you. Here's the whole carpel here, the stigma, and then down to the ovary. Down to the ovule here. And in the ovule, there is an egg cell. That egg cell in the angiosperms undergoes a complex set of divisions. It's a combination of a meiosis and then a mitosis. And we're going to end up with 8, count them, 8 nuclei. All of them haploid. So here we go, here, we count them up. 1, 2, 3, 4, 5, 6, 7, 8. So 8 of them, 8 nuclei. There are only 3 of them that we're really interested in and that we're going to follow. The others form these kind of supporting cells. There's 3 up there and there's 2 down here. We won't include them in our story. Okay, just remember that they're there. The ones that we are interested in are these 3 nuclei. These 2 here and this one here. This nucleus here forms the egg. This cell up here is very interesting because it has also divided by meiosis and then a mitotic division, and it has 2 haploid nuclei, 1, 2, but it does not develop a cell wall. Instead, the cell here has 2 haploid nuclei, and they stay apart from one another for the moment. So when fertilization occurs, the pollen grain lands here, and the pollen grain, the cell of the pollen grain, germinates in a pollen tube. And the pollen tube grows all the way down the stigma, all the way around, and then into the ovule here. And remember that in the angiosperms, the pollen grain produces 2 sperm, and the sperm actually move down and fertilize this egg. But how many sperm do we need to fertilize the egg itself? We only need 1 sperm, and there it goes, and it fertilizes the egg, and we now have a diploid zygote. But we've got another sperm. What happens to it? Well, it also fertilizes, but what it fertilizes is this big cell here with its 2 haploid nuclei, and it ends up there. We end up with 3 haploid nuclei, 3N. Our cell ends up being 3N. That is called a triploid cell. And triploid cells in plants are actually relatively common. In fact, whole triploid plants occur quite regularly in the plant kingdom. They are used. And I'll give you, oh, sorry, this happened again. I think there's something wrong with my speaker. Anyway, we'll just move over here again. Okay, so some plants, I'll give you a good example. The bananas that we eat, the commercially available bananas, they are triploid. They are sterile and they're triploid. It is typical of these triploid cells that they are very large and they accumulate a lot of nutrients. Well, that's exactly what is going to happen with this cell here in the egg. I mean, associated with the egg. This is going to form a very nutrient-rich supporting tissue for the developing embryo. And this is called the endosperm. This fertilized egg here is going to form an embryo, and we're going to see that it develops a particular structure. There are 2 major groups in the angiosperms, and the structure differs a little bit between the 2. But whatever they do, this endosperm here develops, and that is often what we see as the early on. That is what we see as the predominant nutrient tissue for the developing embryo. Things may happen in development a little bit later as the endosperm gets used up, but the endosperm provides the early nutrient to the developing embryo. So double fertilization. Remember, there is a meiotic and mitotic division in the initial spore, in the megaspore. We end up with 8 haploid nuclei, and 2 of those form this endosperm cell here and are fertilized by one of the sperm from the pollen grain. The other sperm fertilizes the egg cell itself, and we end up with a diploid embryo and triploid endosperm. And this is called double fertilization. And it is a major advance, evolutionary advance in the angiosperms. And the reason for that is that it allows for the development of a very, very elaborate supporting mechanism for this embryo. Okay, so just to run through the life cycle of it's basically going to pretty much repeat what I've told you. But mature flowers, what we're looking at, that is a sporophyte. And they produce a megaspore, which is in the ovule. And that is going to, and that's the one that undergoes meiosis and mitosis to produce the 8 cells. And the male parts of the plant, the anther is actually the microsporangium. That is, it is producing microspores, and the microspores mature as pollen grains. They have 2 cells inside, which form the sperm cells. So the pollen grains in general must be transported to the female. In many plants, the pollen grains are so small they are blown by wind. In others, they require transport usually by an animal of some sort. Usually an insect of some sort, but other animals may also be involved. For example, there are desert cacti, which are fertilized by bats. But insects are the most important of all of the pollinators. And many, many insect groups have evolved in very close association with plants. And the two have co-evolved so that the method of fertilization is ensured as long as the insect partner is present. So there's a very close association, a co-evolution between pollinators and the source of the pollen, which is the plant itself. Now, I emphasize as well that the reason that insects go to plants depends upon the plant. The plants can offer various things. First of all, they advertise. That's why flowers have their form, their color, everything else. They advertise to their insect pollinators. But they provide products for the insect. They provide nectar. They sometimes provide things like wax to the pollinator. And sometimes it's pollen itself that the insect collects. Bees, for example, collect both nectar and pollen from the plants to use as a food source. The plant doesn't mind because, in fact, you need very little pollen to fertilize another plant. And as the insect moves from plant to plant, as long as it is carrying some pollen with it, it will affect the pollination. Okay, so let's have a look at the female. We've discussed already, undergoes meiosis and then undergoes a mitotic divisions to produce those haploid nuclei. All the different kinds of cells that we discussed in the previous slide, sitting here inside the ovule and waiting now for fertilization. The pollen tube, the sperm fertilize the egg to provide a zygote, and the endosperm to provide a triploid endosperm cell, which rapidly divides to provide an endosperm to support the early embryo. So the endosperm actually may become used up. If you look at a mature seed, you may not see any endosperm, but in some seeds you do. And for example, let's contrast a bean seed. When you look at a bean seed, the endosperm has all been used up. What you're looking at in the bean seed is effectively the embryo itself. And all the nutrients, everything has become concentrated in the first embryonic leaves of the embryo. That's what you see in a bean. You split it apart. In other things, though, the endosperm is preserved. And one of the most familiar to you will be popcorn. A corn kernel, what you're actually looking at in a corn kernel is the endosperm. There's a very small embryo in the kernel, but the bulk of the kernel is in fact endosperm. When you pop it, what's happening is you're causing steam to develop in all of the endosperm cells, and that's what pops it to make it popcorn. Okay, so after fertilization, depends on the plant. The fate of the whole gametophyte depends upon the plant, but ultimately this seed is formed, maybe enclosed in a fruit, but ultimately the seed itself has to germinate to provide the new sporophyte. Okay, just this is also an extra slide that I've put in here, just to emphasize how remarkable this process of fertilization is. Let's have a look at corn, because corn, each of these here is an ovule there. It's actually a modified, they're actually really, you can think about them as being individual modified flowers. Obviously, they don't have petals, whatever. But what you're looking at in the kernel here is an ovule there. And each ovule has attached to it this long, long stigma, which is what we see in corn as the silk. Now, that can be very long. And the reason is, just think about the kernels down here. Okay, they're sitting down here. They've each got their own stigma, their own silk. It extends all the way out here. Corn is wind pollinated. It produces abundant pollen from a set of male flowers, and that lands on the stigmas. And the pollen tube has to grow all the way down. It has to grow all the way down to the ovule in order to fertilize. And that then develops. And what, as I told you, what we're seeing here in a mature kernel, you're seeing rich, thick endosperm. That's what we eat. Okay, so the angiosperms only began they originated about 140, 150 million years ago, but they only began to be dominant less than 100 million years ago. And really, their explosion as the dominant vegetative form is a relatively recent event. And that very rapid evolution has largely been driven by their close interaction with their pollinators. They have developed very skillful, very, very efficient pollination mechanisms. Some of them do rely on chance, on wind pollination. But many, many, many of the angiosperms have developed these close relationships with insects, and their pollination is extremely efficient. And that is one of the reasons for their great success. Nonetheless, despite the fact that they are relatively recent, their exact evolution, their relationships to one another are not always clear. There are some things which are very obscure about the angiosperms, about the flowering plants, and how they relate to one another. We think we know which are the more primitive, which can be regarded as being more primitive in that they probably resemble the early ancestors more than more recent forms. But even there, it sometimes is not very clear. Plants do appear in the fossil record. There are very few fossils of flowers themselves because, of course, they are so delicate. There are some. It's usually the leaves and stems and everything of plants that will fossilize. There are a few flower fossils, though, which give us a clue as to what the evolutionary path has been. So the divergence of gymnosperms and angiosperms was quite early on, about 305 million years ago. But the angiosperms remained a small, very obscure group until about 140, 150 million years ago. And so we now see about 250,000 living species, and more are being discovered daily. This is one of the reasons why the loss of areas like the
Amazon rainforest, and that is so tragic, is because in that loss, we're also losing species of plants that we currently know nothing about, and we never will. Now, this is a fairly old classification of the plants. Is now molecular studies have kind of cast doubt on this, but it is a useful way for us to kind of organize what we're thinking about. We used to place the more primitive angiosperms in groups which were called the basal angiosperms, and these are strange plants which bear a close resemblance very often to fossil forms. And included in the basal groups, or included with the basal groups, also a group which you'll know very well, and these are the magnolias. The magnolias are regarded as being one of the more primitive angiosperm groups, and for reasons that I'll show you in a second. Apart from those basal groups, which are relatively, I wouldn't say they're unimportant, because of course they're important, their very existence is important, but numerically, in terms of species numbers and everything, they are of lesser importance. The two major groups that have evolved in the angiosperms is a group called the monocotyledons and the dicotyledons, or now referred to as the eudicotyledons. And I will still make the mistake and call them dicotyledons because I'm old. And it takes me a long time to get my brain to say the new things that I'm supposed to say. So the old names were the monocotyledons and the dicotyledons. So I need to just explain to you what monocotyledon means and what dicotyledon means. The cotyledon is the earliest embryonic leaf, and it forms in the embryo. And in the monocotyledons, which typically things like grasses, palms, orchids, these things are all monocotyledons. There is just one of these leaves. In the eudicotyledons, the dicotyledons, there are two of them. And just think back what I just told you about. The bean seeds, for example. When you split a bean seed, right, it splits into two. Each of those is a cotyledon. You've probably grown beans. As a kid, you probably grew beans, damp tissue paper, whatever. And when the first thing they do, they put up these two fleshy, fat leaves. Those leaves, cotyledons, are filled with nutrient, which they've derived actually from the endosperm. But the cotyledons can become photosynthetic. So they are the first leaves. They're the embryonic leaves. One in the monocots, two in the eudicots. All right, let's have a look at some of the more primitive ones. The only one I think which I'm going to discuss are the magnolias. The group is called the Magnoliids, and they are neither monocots nor dicots. They sit somewhat outside. And to tell you the truth, their exact affiliations are not clear even on molecular study. But we suspect that these are close to a primitive or ancestral form because we see magnoliids early on in the fossil record, relatively early on in the fossil record of flowering plants. And this is what a magnolia looks like. First, I'll tell you what the primitive things are. First of all, they have many petals. We'll call them petals. They have many petals, which is a primitive feature, and the petals are arranged in a spiral, which is not true of the more advanced angiosperms, where they are arranged in whorls, in circular things below one another. The second thing is that the magnolias actually have a cone, which rather resembles that of the gymnosperms. And here is a mature magnolia cone. Each of these here is an ovule there. So, and again, these cones are arranged in a spiral, which is typical of the gymnosperms. So, now I'm not saying that these are gymnosperms. I'm just suggesting to you that they are preserved. There may be here preserved some gymnosperm features of the early evolution of these angiosperm groups. You'll know most of the magnolias that we have in the United States produces beautiful, big tree with these magnificent flowers, especially this time of year. The magnoliids actually are quite diverse. There are some smaller plants. And in fact, one of them, our most useful plants, is a magnolia, and that is the pepper. Black pepper is actually a vine, a climbing vine, but it is a magnoliid. So that's one of the primitive groups. There are some other more primitive groups as well, but we won't worry about them. So now let's talk a little bit about the monocotyledons. Monocotyledons, they have one embryonic leaf in the embryo. They are also a very, very diverse group. And you'll see many, many, many strategies for pollination in the monocots. The most familiar ones to you, first of all, will be the grasses. These and the grasses, it's hard to believe that they are a flowering plant, but they are. They have a very reduced flower, and here is the structure here. There's in general, there's some variability. There is an ovary, and their male parts, stigma, the anthers, and then some, usually a filament and an anther, and then usually stigmas leading to the ovary. And as I've told you, we saw earlier, corn. Corn is a grass. It is one of the grasses, in actual fact, and it basically has the same structure. It has each of the kernels that you see. It consists of an ovary. The stigmas are the very, very long silks, but the anthers are separate, and the pollen is produced separately on a male stalk. That is quite common in the grasses. This is oat or barley. You can see that they have very reduced, they're not petals, but they have a very reduced outer covering to the flower. The palms, all of those trees, they are all monocots as well. And so are the lilies and things like that. And in general, what you will see in the monocots is that the petals and sepals of the flower, if they have a flower, the petals and sepals of the flower are in units of 3 or multiples of the 3. So here we have 3 petals there, and on the outside 3 sepals, but they're very, very similar. And they're called tepals because they're indistinguishable from one another. Same thing, I'm sorry to hurry through it. We could spend an entire semester talking about this. The orchids, I just want to spend a minute talking about them because they are so spectacularly specialized. The orchids are monocotyledons. You can see that they're, in fact, this is a relatively easy one to work out. You can see 1, 2, 3, 1, 2, 3. But in the center here, there is a very, very, very highly modified carpel and also anthers, all united into a structure which very often can be fertilized only by one insect, by one particular insect. The orchids specialize in co-evolution with their pollinators. And very often it is so intense, the co-evolution is so intense, that the two partners become absolutely dependent on one another. If you take the orchid pollinator and render it extinct, the orchid will go extinct. If you took the orchid and made it go extinct, the insect pollinator would go extinct. It is that intimate. I won't discuss all the mechanisms, but if you are interested in this, read up a little bit about orchids and how they are pollinated, because their pollination mechanisms are quite spectacular. Okay, so these are eudicotyledons. Now, eudicotyledons also, remember, they're flowering plants, and the flower is a major part of their evolutionary strategy for attracting pollinators. And in the eudicotyledons, the floral parts are usually in multiples of 5. But there's a tremendous amount of variability in how that is done. In this one here, you can see this is a dog rose, an uncultivated rose. 1, 2, 3, 4, 5, and we'll find that the carpels are in units of 5 as well, and all the male parts will be multiples of 5. And in both monocots and eudicots, the floral structures are arranged in whorls. They're not arranged in a spiral like they are in the primitive flowering plants. Nonetheless, in the eudicots, there is a huge variability in the structure of the flower, and some of them can be quite specialized. But I don't think anything approaches this kind of specialization. There may be one group in the eudicots that does, but the monocots seem to, in the orchids in particular, are the most spectacular examples of co-evolution. The eudicots are predominantly insect pollinated, but there are some, like the oaks, for example, and large eudicot trees are mostly wind pollinated, not insect pollinated. The typical groups in the eudicots, things like legumes, squashes, those sorts of groups. The flowers are not always symmetrical. They're not, sorry, excuse me. They are not always radially symmetrical. Very commonly they are. All of these are radially symmetrical, but they may be bilaterally symmetrical, as they are in the legumes. Okay, so just to emphasize, these two groups, the monocots and the eudicots, appear to have diverged from a common ancestor, although some people would argue about that, even about that. But when we look at the modern monocotyledons and the modern eudicotyledons, we see some very, very distinct differences between them. Not only in the flowers, as I've told you, but in many other parts or structures of the organism. Just excuse me 1 minute. I'm just going to pause for 1 second because I need to just go and sorry, I got something stuck in my throat. So, okay, not only in the flower structures are there differences between monocots and eudicots, but in many other aspects of their morphology, of their plant anatomy. So first of all, we distinguish them immediately on whether or not they have one cotyledon or two. That's where the name comes from. So in the monocotyledons, there is a single embryonic leaf. In the dicotyledons, there are 2 embryonic leaves. And remember, this is embryo, right? We're not talking about the endosperm, we're talking about the embryo itself. Second of all, in general, there are 1 or 2 exceptions, but in general, when you look at the leaves, in the monocotyledons, the venation, the veins you see are parallel to one another in the monocotyledons. Just think about a grass blade, right, or a corn leaf, and you'll immediately be able to visualize these parallel veins running through it. In the eudicotyledons, that is not the case. The veins are usually a net-like, or radial, they spread out from one another through the leaf. As I say, this the word is important usually because there are one or two exceptions, which are just specializations of the particular species. This won't mean a lot to us at the moment because we haven't talked much about the vasculature of plants, but I will just explain to you that the vasculature, the tubes which conduct fluid in a plant, are organized into a particular structure, into bundles called the vascular bundles. And when we look at monocotyledons, the vascular bundles scattered right throughout the stem. They look almost random throughout the stem. When you look at the eudicotyledons, they are very strictly organized into a ring in the stem. Now, this has huge implications for the plant. And the eudicotyledons in general are regarded as being, in many ways, much more successful than the monocotyledons in terms of their diversity and size of the stem, because this allows a particular kind of growth. And the monocotyledons have great difficulty in developing very large plants because these scattered vascular bundles impose a particular limitation on them, which we'll discuss if we talk about the vasculature. This ring structure allows a plant to grow in width quite easily and to build very strong tissue in the center. So all of the woody, truly woody plants, the plants that we derive useful wood from, are eudicots, because only the eudicots that can make true wood. And that's a huge evolutionary advantage. It allows them to become enormous. So the next thing is that here's another usually. Usually the monocotyledons have a root system which is fibrous. They don't have a taproot that goes down. They have a net-like structure of roots that spread through the soil. There are one or two where it begins to look like they have a taproot, but that is a specialization. Again, in the eudicots, their root usually grows by putting down a central root or a taproot from which branches emerge. But again, there are some which are specialized, where the taproot just divides very, very early on and it looks like this. But in general, monocotyledons have a net-like root structure and the eudicotyledons have a taproot. When you look at the pollen of monocotyledons is different in structure to that of the eudicots because the pollen has one small opening here, it has a pore in it, where in the eudicotyledons, there are 3 different openings. Sorry, just go back here. Okay, and then the last major difference that we'll note between them is what I mentioned before. The floral organs in the monocotyledons are usually organized in multiples of 3. We'd see extensive modification of that. For example, we've already talked about the grasses and things like that, where the petals, sepals, everything might be lost entirely. And there are others where the structure of 3 has become so highly modified that it's actually quite difficult to work out how many they have. But in general, if you examine the development of the flower bud, for example, you can see it quite easily, even in very specialized monocotyledons, that their floral organs are organized in units of 3, and that is all of the floral organs. The sepals, petals, the female carpels, the number of filaments and anthers, etc., etc. It's all in multiples of 3. In the eudicotyledons, they are organized in units of 5 usually, but sometimes of 4. There are a couple of very big eudicotyledonous groups where it's organized as 4, but it's never as 3. Usually it's 5. Okay, so let's just have a look. I just want to see. Okay, so just to remind you, here are the things, the derived traits of seed plants, or these are things developed early on in the ancestry of the seed plants, which now characterize the entire groups. Highly reduced gametophytes of both male and female, but the gametophyte is still there. It's just that now it's microscopic, not a large gametophyte like we saw in the primitive plants. The second thing is there is pronounced heterospory. That is, there is a very large, comparatively large female megaspore, which gives rise to the female gametophyte. And that is by those divisions that we've talked about already. The microspore is male, and that is the pollen grain. And inside the pollen grain is the male gametophyte, which produces the 2 sperm nuclei. The next, this is the thing that really distinguishes the seed plants. Sorry, let me phrase it this way rather. The seed plants have the characteristic that they all have ovules, but this is the one thing which distinguishes the angiosperms from the gymnosperms. In the gymnosperms, the ovule is not enclosed in anything. In the angiosperms, the ovule is enclosed in an ovary. So the gymnosperms, the ovule is naked. In the angiosperms, it is enclosed in an ovary. Nonetheless, the ovule is a typical derived trait of all of the seed plants. Pollen is apart from the development of the seed, the evolution of pollen was one of the most important traits, derived traits, which allowed plants to penetrate onto land because it freed plants from the requirement for water in order to effect fertilization. In all of the primitive plants, the non-seed plants, the sperm have to swim. They have to actively swim towards the archegonium, and they swim through water. They have to swim through standing water. In the seed plants, that is no longer necessary because the pollen actually germinates a pollen tube through which the sperm move to the egg. And the last thing, of course, the major derived trait is the seed itself. The seed is the embryo protected in some way by a coating, by the integument. And the seed itself allows for resistance to desiccation, for dispersal, all of these advantageous traits. In the earlier primitive plants, in general, the spores were unprotected. and at the mercy of the climate, etc. Seeds can remain dormant until such time as conditions are suitable for their germination and for the growth of the plant itself. Okay, so that's it for today. And then our last classes, we will be discussing plant vasculature and how plants actually move food and water around. Okay,